Good morning, everyone. Today, I am presenting our tool IdentifiHR, a model for predicting homologous recombination deficiency in high-grade serous ovarian carcinoma through gene expression. So I want to start by giving you some insight into our motivation for actually developing IdentifiHR. Approximately 50% of high-grade serous ovarian carcinomas have a therapeutically targetable defect in DNA repair, or more specifically, in the homologous recombination repair mechanism for double-stranded DNA breaks. While homologous recombination, or HR proficient cells, are able to repair double-stranded damage through this relatively error-free mechanism, those that are deficient are unable to do so. And this leads to an accumulation of damage and scarring in the genome. However, these scar patterns can be summarized as a genomic HRD score derived from SNP arrays, whole genome sequencing, and whole exome sequencing, and form the basis for homologous recombination status classification in cancer, being HR deficient or HRD and HR proficient HRP. And this is really important clinically because the HRD high-grade serous ovarian carcinomas are eligible for targeted therapy. So we need to pick up all cases that are HRD. Despite, however, the clinical importance of this molecular feature, there are currently no methods to predict HR status using gene expression in high-grade serous ovarian carcinoma. And so naturally, we set out to build one. To do so, we took the Cancer Genome Atlas, or TCGA, cohort of 361 high-grade serous ovarian carcinomas, and we randomly split it into training and testing sets. We then annotated truth labels to our data using this gold standard for classification, being the genomic HRD score, and accessed bulk RNA sequencing for just over 52,000 genes per sample. This is a very large feature search space, and so we wanted to begin by reducing it through manual filtering and then through filtering by differential gene expression analysis, whereby we contrasted the cases that were HRD against those that were HRP using the standard Limma-Voom pipeline. And when we set our significance threshold at 0.05, we were able to discover 2,604 significantly differentially expressed genes. Now, this is great, but we want to make sure that these genes are actually giving us some insight into our homologous recombination deficient phenotype. And so what we looked at, in addition to the usual sort of volcano plots and whatnot, were the frequency of our significantly down and upregulated genes based on their chromosome. And what we were able to appreciate was that some chromosomes were completely enriched, seemingly, for just downregulated or upregulated genes. So we probed this result a little bit further. And so there should be many gray dots here, but they're gone, so you're going to have to imagine them. When we plotted the location of all of the genes profiled by differential expression analysis against their significance, coloring by whether or not they were significantly down or upregulated, we were able to observe that differential expression seemed to be occurring in banded regions. So regions of the genome would be purely down or upregulated. And so this, to us at least, implied potentially that our differential expression signal was actually picking up copy number alterations. And so we did a bit of a literature search, and we're pleased to see that there are known copy number alterations that occur specifically with HR deficient or HR proficient statuses. So HR deficient high-grade serous ovarian carcinomas are known to have a copy number amplification, for example, at chromosome 8's Q arm at band 24. And sure enough, we see an enrichment for just upregulated genes in this region. So our gene expression signal is capturing a genomic sort of copy number signal as well. And so given this and the fact that the copy number signal has proven to be a robust way of classifying this phenotype historically, we proceeded with these DE genes. We used them as input when training a penalized logistic regression model. We tuned our L1 and L2 hyperparameters through 5-fold cross-validation, and taking the average values that gave us the maximum area under the curve, what we were left with was an elastic net penalized logistic regression model of 209 weighted genes that would hopefully be able to predict HR status in high-grade serous ovarian carcinoma. And we called this method IdentifiHR because I was allowed to name it, and so that's what I chose. We have built IdentifiHR as an R package, and to use it, all an individual needs, all you need if you wanted to use my package, are just raw gene expression counts for either a single high-grade serous sample or a cohort of samples. And what the model will give you is a discrete HR status prediction, HRD or HRP, in addition to the probability of a sample being HRD. So it's great that we've trained this model, but we also need to prove that it works. And so we went back to our TCGA cohort and accessed our held out testing subset. And when we ran the IdentifiHR model to predict HR status on these high-grade serous cases, we were pleased to see that we had an accuracy of 74% and an area under the curve of 0.85. What we were also pleased to see is that when our model incorrectly predicted a class, there was a fairly even balance between these incorrect predictions. So the model wasn't preferentially calling all samples a particular class, which was quite nice to see. What we were more interested in, I know accuracy is very important, but it's also important that we convince clinicians and other scientists to use this tool, was the correlation between the probability outputted by our model and the genomic HRD score, which is the gold standard and which forms the basis for our truth labels. And so we assessed the probability of a sample being HRD as outputted by IdentifiHR against the genomic HRD score. And we were pleased to see that they were strongly positively correlated, and where IdentifiHR was incorrectly predicting a label, the probabilities were quite close to the threshold set. We then wanted to benchmark IdentifiHR against existing homologous recombination gene expression signatures. Now, to our knowledge, there are none for high-grade serous ovarian cancer or developed specifically for high-grade serous, though there are two that were developed using the TCGA breast cancer cohort, being the BRCA signature and the multiscale HRD signature, and one developed in a TCGA pan-cancer cohort called expression HRD. And so when we plotted the 73 high-grade serous ovarian cases from the highest HRD score through to the lowest, also annotating their genomic homologous recombination status, what we were able to appreciate was that IdentifiHR correctly predicted the most, or the labels of the most samples, as opposed to BRCA multiscale HRD and expression HRD, meaning we had the highest accuracy and precision. And while expression HRD did beat us in terms of recall, that was because it called everything HRD. So we were happy to let them have that metric. We also had the lowest misclassification error. When we looked at the overlap between the features for these four signatures, if you will, or models, we did observe that multiscale HRD and expression HRD did have some overlap with our features, and that only one gene overlapped between all four models. And it's actually quite an interesting gene, which I won't be speaking about today, so you're going to have to come and talk to me at my poster. What's more important in terms of this work is that our model actually generalizes to independent data sets. And so we were really fortunate to have access to 99 samples collected from 81 patients of the Australian ovarian cancer study. We used IdentifiHR to predict HR status in these samples from gene expression, and we had an accuracy of 91% and an area under the curve of 0.95. But what's really interesting about this data set and these samples specifically was that they were derived from different tissue types and at different time points. So while we did incorrectly predict the label of 7 of our primary high-grade serous tumors, we correctly predicted all of those collected after autopsy. We correctly predicted all of those taken from ascites, which represents a promising liquid biopsy alternative for high-grade serous. And we correctly predicted all of those from normal fallopian tube, which represents the tissue of origin for this disease. So this was quite a nice result and gave us maybe a little bit of confidence in our model. And so despite only training on bulk RNA sequencing data, we then undertook testing of IdentifiHR in pseudobulk high-grade serous cells from 106 samples taken from 37 patients of the Memorial Sloan Kettering Cancer Center. We subset our cells down to just high-grade serous cells, so just tumor cells, and pseudobocked at the patient level, and we're pleased to see that we had an accuracy of 84% and an area under the curve of 0.92. So despite training on bulk RNA sequencing data, we've proven that we also work in pseudobulked cells, and more so that our signal is high-grade serous cell tumor specific because we've removed the microenvironment in filtering other cells. So to summarize, we have trained and tested a model called IdentifiHR. We've benchmarked this model. We've tested it in independent cohorts. There are a range of other really interesting smaller findings. So if you would like to hear more, please come and visit me at my poster. I'll also be presenting later in the week. Check out our preprint. When the paper's published, cite us, please. Yeah, thank you.
So I have one question actually. So you trained a model. So how many number of features were there? Like because it's gene expression, so the genes will be features, right?
Yeah. So we started with 52,000 genes, and then we manually filtered, and what we were left with were 209 weighted genes. So there are only 209 weighted features in the model, but there are 2,604 features that are used for normalizing library size differences. That's the sneaky bit. So you do need 2,604 features to use.
Yes, exactly.
Genes to actually run the model. Yeah.
And same thing, because you're predicting something, right? So you need the same features. And it is like very rare that you will get the same features when you're predicting some other sample as well. So how do you tackle that?
So we tried to overcome this issue when we were actually beginning our filtering steps. So we filtered any genes that were non-coding because they're not as well annotated, even though they're very interesting. We filtered genes on Y chromosomes because they're not relevant for this disease. We filtered genes on mitochondrial chromosomes because they tend to be a little bit more variable, or at least that's what the literature suggested. And we filtered any that were just broadly lowly expressed because we assumed that they wouldn't contribute to the prediction anyway. And then we performed differential gene expression analysis. We kind of checked our genes, the right genes were coming up. Yeah, it's very difficult to ensure that future data sets have all of these genes. However, when we performed our testing in our pseudobulked single cell cohort, there were 8 out of our 209 weighted genes that were actually missing. Now, their weights weren't huge, but we ended up just sort of setting their values to 0, and we still had quite a high accuracy. So we're happy with our features. It's being reviewed currently, so they're not changing. But hopefully it is accessible and usable. Within the package, we do have functions for users to be able to interrogate missingness, if any, in their data set to kind of visualize the weights of different features and how that then might have implications for model predictions as well.
Okay. We can have more discussion in the poster halls because we are having limited time. So it's fine. Thank you so much. We can have